Generally, electrophoresis can be used to visualize bands of a molecular marker to genotype individuals, verify amplification by PCR and sequencing reactions, check the quality and quantity of genomic DNA after an extraction, separate DNA fragments to clone a specific band, or separate complex mixtures of proteins from cells, subcellular fractions, column fractions, or high-immune precipitates for investigating subgenic compositions and to verify homogeneity of protein samples. Electrophoresis has several requirements according to the loaded sample. For DNA, the gel matrix composition is comprised of agarose and a running buffer. For RNA, you need just agarose and water, and for proteins, you need a acrylamide and bisacrylamide matrix with 4x3Cl, APS, and T-mid buffers. The running buffer for DNA is comprised with 1xDA and or TB buffer, and for RNA you need a 1x-MOPS buffer, and for proteins a 1x-SDH buffer for both running and loading buffer. But for DNA loading buffer you need a 6x loading buffer, and for RNA a 10x MOPS running buffer. Then for power or running conditions, you need a 10 volt per centimeter for DNA samples, and for RNA, just the half of the voltage. For protein, you need 10 to 15 milliamperes, and for visualizing DNA, you need ethidium bromide and ammonium acetate, both for DNA and RNA, and for proteins, there are several staining processes like QMSI blue, silver staining, fluorescent protein gel stains. Here are some steps for performing nucleic acids electrophoresis. First, prepare and powder the gel. This will take about 45 to 60 minutes. The second step is to prepare and load samples. This will take you 10 minutes. The third step is to run the electrophoresis gel, 45 minutes to overnight, and the last step is to stain and photograph the gel. About an hour you will take.
Electrophoresis of DNA at different concentration of agarose in buffer TBE 0.5x and TAE 1x, gel A 0.7% of agarose in buffer TBE, gel B 1.5% of agarose in TBE, gel 2.5% of agarose in TBE, and gel D 1.5% agarose in TAE. All the gels were loaded with the same sample to compare migration in each one. Number 1, 1 kilobase marker oxygen 5 microliters, PCR sample and loading buffer at 6 microliters, DNA sample and loading buffer 6.5 microliters, and for plasmid and loading buffer 7 microliters. All the gels were strained with bromophenol blue and raised in 120 volts and 45 minutes. Sample A 0.7% agarose TBE buffer with shift a longer DNA sample shown. Sample B 1.5% TBA buffer shows in line 4 as amino shift much of the sample is near the wells, while PCR sample is in line 2 doesn't run as far as image A. Sample C, the plasmid, the PCR product, and the marker run on the health viewer A and B. This is because the pure size is smaller because the agarose percent increase. The later sample was made with TA buffer, easy to avoid for low voltage, short shift. The difference between TBA buffer and TA buffer is that it is adequate for a smaller sample of DNA. Square yellows we have the white color displayed excess on dye in the loading buffer. Square purple it shows the wells is because there is presence of proteins. The squares in blue is relaxed plasmid DNA. Circle blue, this strain is because the formation of bubbles within the basin transmillimeter. Square in green, supercoiled plasmid. Square in yellow is genomic DNA. And finally we have square in red, that is degraded DNA. This is maybe because the sample is all the time shift and the voltage were not optimal for the proper shift is needed to a voltage of 5 volts per centimeter. Therefore, had to be run at 100 volts for 60 minutes.
By observing the four agarose gels, Gel A shows the better results for DNA lather. It can be distinguished each mark of the 1 kilobase oxygen lather. As for PCR sample, on pit number 2, it can be clearly observed the migration in each of the gels. No degradation is observed, but an excess of salt on gel B and probably a contamination on gel A, white spots were spotted. About genomic DNA, on pit number 3, on every gel degradation is observed. Degradation of genomic DNA may be caused by sample age and also because of non-optimal running conditions. Although the better results for this sample were shown on gel A, this may be explained due to the fact that TBE buffer is better for running small samples of DNA. Also, salt success are shown in every gel. While on every pit number 4 of gels, it can be observed contamination with proteins, degradation, and possible salt excess, being gel A the most representative of all. Depending on the size of the sample is the agarose percentage recommended. The general rules goes by the fact that increasing the concentration of agarose, the size of pores are smaller, therefore smaller fragments of DNA can be separated. The running buffer was selected because of characteristics of the sample. 0.5x TBE buffer was used because of the size of the DNA lather. Also, this buffer has greater regulation capacity than Ti. It can be running for longer periods of time without the fear of acidification of medium. About the loading buffer, if you're expecting a genotyping band of 200 to 400 nucleotides, you shouldn't use bromophenol blue since it will obscure your product. In this case, you should use a larger dye like hyaline cyano. As for the use of syrup safe, it's marketed as being less harmful than ethidium bromide. Still, other manufacturers claim this to be debatable. Nevertheless, its major advantage, among other stains, is that it is as sensitive as ethidium bromide, less methygenic, although its toxicity is higher. Its manufacturer claims to be safe for the environment and easy to dispose. Other stains such as methylene blue, crystal violet, or even syrup green, including ethidium bromide, are highly methygenic and hazardous to the ambient. Therefore, gels containing these dyes must be handled and disposed as hazardous waste. The voltage applied was adequate to show proper results, thus making the analysis of this, roughly speaking, easy to interpret and draw conclusions about the samples loaded. If a higher voltage is applied, then the electrophoresis would have been a failure, due to the fact that if temperature within the electrophoresis chamber increases, agarose gel will melt down. Also, if a lower voltage is applied, then the samples won't be able to migrate properly, because the voltage is not enough to cover the whole chamber length. The best condition for this electrophoresis chamber would have been 100 volts for 60 minutes according to chamber length and sample's weight.
To verify integrity of extracted nucleic acid display such materials for electrophoresis technique that is used, sample deposit in the well 3 DNA corresponds to the quality of the sample is not good because it is degraded. In this photograph show sweeper not defined bar is. It should take in account what kind of the gel is used for each sample. Type of refers to the size of DNA because as it was observed depending on the percentage of agarose in the shift of the sample. In order to obtain adequate results, you need to have a well-standardized protocol, respect the voltage and the times for the power supply, as well as sampling from adequate manner to prevent contamination.